Hi everyone. Today the topic of over-discussion is inverse PCR. In this video, I will discuss rationale, principle, how this PCR works, add in the end advantages and disadvantages as well. Let's see the rationale first. If you have an unknown species, but you have a short-known gene sequence that may occur in that unknown species, you should use inverse PCR and get the whole sequence easily. If we look at the principle, it is in PCR in which, on the basis of a short-known sequence, we find out the sequence of flanking region by reversing the orientation of primers. There are few important points as follows. Short-known gene sequence will be used as start point. Transposable elements or gene can act as a start point as well. Now, if we look at how it works, this PCR can be divided into following major step. Step I, initial restriction. Digestion. Step 2, ligation. Step 3, second restriction. Digestion. Step 4, normal conventional PCR. For the amplification, consider a genomic DNA region with known sequence, and you want to determine the sequence flanking it. For this, a primer pair designed from known sequence region but orienting outwards. Next, you cut the large genomic DNA using restriction enzyme such that you have a small amplifiable fragment a few kilobases long. This fragment is circularized and ligated using DNA ligase. A second restriction enzyme cleaves within the known DNA sequence to yield a linearized DNA fragment with unknown sequence at the center and known region flanking it. You can see the primers that were signed orienting outwards are now pointing inwards. Now, standard PCR amplification can be performed using a thermocycler. Now the advantages are, at least we have the start point. We can get the sequence of flanking regions. And disadvantages are as follows. Enzyme-dependent reaction has high failure chances. Recognition site is hit and trail method costly time consuming. Thanks for watching, and don't forget to subscribe our channel.